Hello, good afternoon. Thank you for joining us. My name is David Taylor. I'm an R&D scientist from Cayman Chemical. I'm going to be giving a short talk about modulation of ENPP1 activity in ovarian cancer cell lines via loadable lipid nanoparticles. Lipid nanoparticles are a class of nanoparticle that are able to encapsulate nucleic acids such as DNA and RNA. We've been working on a line of products that we call loadable LNPs. Now, in conventional lipid nanoparticles, the cargo is encapsulated at the same time that the LNP is formed. In contrast, loadable LNPs are a preformed lyophilized empty LNP. The reagent is rehydrated, mixed with RNA cargo, and then diluted in an encapsulation buffer to complete loading. Loadable LNPs exhibit comparable RNA encapsulation efficiencies to conventional LNPs and exhibit consistent sizes with conventional LNPs in the 80 to 125 nanometer range. We've been testing loadable LNPs in ovarian cancer cells and macrophages, looking to modulate ENPP1 and STING activity. Now, the cGAS-STING pathway regulates activation of innate immunity in response to misplaced DNA, which can arise as a result of infection or in the setting of cancers. cGAS recognizes DNA in the cytosol and generates the second messenger, cyclic dinucleotide, 2,3-cGAMP. 2,3-cGAMP is a second messenger which is then exported out of the cell and is able to bind STING in target cells, which then leads to stimulation of type 1 interferon response and stimulates antitumor immunity. Cancer cells can also express a protein called ENPP1, which is a membrane-bound cGAMP hydrolase. ENPP1 can degrade cGAMP, and this leads to immunosuppression via preventing STING activation, but also through increasing adenosine levels, which is produced from one of the degradation products, AMP. And ENPP1 is frequently upregulated in cancer cells, which can contribute to immunosuppression in tumors. In this study, we sought to modulate ENPP1 activity and expression in ovarian cancer cell lines via loadable LNPs. We tested whether loadable LNPs could be used to deliver cGAMP to THP1 macrophages, and we also investigated whether ovarian cancer cell-derived EVs can impact STING signaling. We first transfected ENPP1 siRNA with loadable SM102 LNPs into 3 ovarian cancer cell lines, SCOV3, PA1, and CAOV cell lines. We observed 50 to 90% transcript knockdown, and this was confirmed via Western, though protein levels in the CAOV3 cells were not detectable via the Western. We can monitor ENPP1 activity using this fluorometric assay, which takes advantage of this non-fluorescent substrate Tokyo green MAMP, which is cleaved by ENPP1 to generate Tokyo green. As you can see in this assay, we were able to confirm significant knockdown in the siRNA treated condition, and this activity was inhibitable with small molecule inhibitors. The assay was sensitive enough to confirm knockdown in all 3 cell lines, even in the CAOV3 cells where the expression was very low. So we then sought to determine whether ENPP1 silencing can lead to changes in cGAMP levels in the media. We used an ELISA to measure cGAMP levels, and we were initially unable to detect cGAMP in the media in any of the 3 cell lines if FBS was present. We then switched over to serum-free culture when we were able to detect cGAMP produced only by the PA1 cells. It wasn't detectable in the media from the other 2 cell lines. And we observed that in the SI-ENPP1 treated condition, there was more than a 2-fold increase in cGAMP levels, indicating that silencing of ENPP1 gave rise to increased cGAMP export by the cells. At the time that we performed these experiments, there was one report in the literature describing a role for ENPP1 in exosomes, specifically in the context of bone mineralization. So we asked whether cancer cell-derived EVs may contain ENPP1. So we isolated the EVs from the conditioned media from the 3 different cell lines. We saw that there was a strong ENPP1 expression in the PA1 cell-derived EVs, which was interestingly significantly reduced in the ENPP1 silence condition. We weren't able to detect ENPP1 in the EVs from either of the other 2 cell lines. We then assayed these EVs in the fluorometric ENPP1 activity assay, and we confirmed that the EVs can hydrolyze the TG-MAMP substrate, and activity was reduced following silencing in these PA1 derived EVs. Interestingly, we also isolated EVs from fresh medium containing either 10% FBS or 10% EV depleted FBS. What we saw was that regular FBS contains EVs which contain a high ENPP1 activity, while in the EV depleted media, the rates were much lower. We then sought to investigate whether these EVs can degrade the natural ENPP1 substrate, 2,3-cGAMP. So the EVs were isolated from conditioned media of the PA1 cells in both the control and silence condition, and then incubated in PBS in the presence of cGAMP. We then measured cGAMP levels via ELISA after a period of time, and we confirmed that the EVs can degrade cGAMP and that the rates of EV-associated cGAMP degradation were reduced in the silenced condition. In the context of cancers, STING activation in antigen-presenting cells promotes tumor immunity. However, uptake of cGAMP may be limited because it's transport protein mediated. cGAMP does, however, carry a negative charge similar to nucleic acids and therefore may be amenable to encapsulation in lipid nanoparticles. We encapsulated Cy5 labeled cGAMP in loadable SM102 LNPs, and we then transfected into THP1 cells and compared with adding the labeled cGAMP directly to the medium. As you can see from the image on the right, we were able to see substantial higher dye uptake with the loadable LNP delivered cGAMP. We then performed essentially the same experiment treating the THP1 cells with cGAMP alone. This time we're using a Western and we're measuring STING protein levels as a surrogate for activation. So loss of STING protein is suggesting activation of STING. When the cGAMP was added directly to the medium, we didn't see any difference in STING protein levels. However, when the same dose of cGAMP was delivered via loadable LNPs, we saw a drastic reduction in STING protein abundance, suggesting activation of STING. We also measured cytokine production by THP1 cells in response to these treatments, and we observed that for the same dose of cGAMP, loadable LNPs were able to affect higher levels of IL-6 and IL-1 beta production. In this experiment, we also co-incubated the THP1 cells with EVs isolated from the PA1 cells. These are the EVs that contain the active ENPP1. When the EVs were administered together with the cGAMP added directly to the medium, we didn't see any change in cytokine release. However, interestingly, when the EVs were co-administered with the cGAMP containing LNPs, we saw this significant reduction in IL-6 and IL-1 beta production. We think this is an interesting observation. We're not entirely sure what it means, but we speculate that there may be an interaction taking place between the EVs and the LNPs. This may be happening in the medium, or potentially it could be happening inside the cell because they could be sharing common uptake pathways. So potentially the EVs and the LNPs are commingling as they're taken up by the cell, which results in a reduction in cGAMP levels actually finally reaching STING. So in summary, here we have our model PA1 cancer cell, which is basally expressing high levels of ENPP1, and it's also exporting EVs containing ENPP1. And this then leads to lower levels of cGAMP and is immunosuppressive. When we silence ENPP1, as you can see on the right, there's a reduced expression of ENPP1 on the surface and also EV-associated ENPP1, and this together contributed to higher concentrations of cGAMP in the medium, which is immunostimulatory. And we also saw that the LNPs can be used to deliver cGAMP to THP1 cells, and this can promote STING activation. However, this was mitigated by the PA1 cell-derived EVs, which we presume to be associated with their ENPP1 activity. In summary, loadable LNPs enabled effective siRNA-mediated ENPP1 knockdown in all three cell lines. cGAMP delivery and STING activation could be enhanced when delivered via loadable LNPs. PA1 cells secreted extracellular vesicles containing active ENPP1, which inhibited cGAMP-induced cytokine release in THP1 macrophages. And finally, we observed that EV-associated ENPP1 may exert paracrine immunosuppressive function in the tumor microenvironment. To conclude, I would like to acknowledge my colleagues at Cayman Chemical, and I'd like to thank you for your attention.